The Autism Research Institute relies on the generosity of donors like you to make this webcast possible. If you enjoy this presentation, please consider making a donation. Thank you.
The link will be in the chat and it was provided in your reminder email. There are no handouts for this webinar. Now, before we get started, I'd like to introduce our speaker. Jamie Dewitt is an associate professor in the Department of Pharmacology and Toxicology at the Brody School of Medicine at East Carolina University. Her current scientific research focus is on how exposure to emerging contaminants in the aquatic environment, such as pharmaceutical and personal care products, affect the adult and developing immune systems. This webinar was made possible through generous donor support, including a grant from Local 25 Boston Teamsters. If you'd like to contribute, please visit our website at autism.com. And now I will turn it over to Dr. Dewitt.
Thank you, Denise. Thank you for inviting me to present a webinar for everyone. I think this is a really amazing platform to bring together people from various communities, from parents to healthcare providers to scientists like me who are trying to understand what environmental contaminants are doing to our health and to the health of our children. Just to give you a little bit more information about what I do in my laboratory here at East Carolina University. I'm an environmental toxicologist, and so most of my work focuses on describing, exploring, and understanding the effects of environmental contaminants on the developing immune system, and then the relationship between the developing immune system and the developing brain. So thank you, everybody, for taking some time out to listen to me talk about some of the work that we're doing here in the lab. I need to say thank you to the hard workers who have put time and effort and thought into many of the projects that we do here in the lab. Ching Hu is involved in almost every project in the laboratory. Sam Vance is an undergraduate student who would come in some mornings and say, "Hey, Jamie, I took care of something for you," and it would be something that I had forgotten about. So he's here at ECU getting a master's degree. And then I want to thank Dr. Anna von der Ems, who is now a postdoc in the laboratory of Dr. Pam Line and Michelle Lamerrill at UC Davis. I'm going to go over a little bit of information related to developmental immunotoxicity and sort of how I think about the developing immune system from the perspective of a toxicologist. A paper came out in 2008 in a journal known as the Journal of Toxicology and Environmental Health, Part B. And this was a review type paper by Dr. Rodney Dietert and his spouse, Janice Dietert. Dr. Dietert is a professor at Cornell University and is one of my research and academic mentors. This particular paper outlined some of the commonalities shared in timelines or critical windows between agents that impact the developing immune system and agents that have been associated with autism in human patients. And so by looking at this relationship, they came up with three main models to help consider or explain how environmental agents can impact both the developing immune system and the developing brain. And I'm going to go through each of these models individually and then take you down the path, as I said, of Alzheimer's disease. So in the first model, the assumption here is that there is a direct cause and effect relationship where a particular environmental agent produces dysfunction directly on the developing immune system, and then that damage in the developing immune system is able to inflict changes in the developing brain. And in some situations, for the right subset of individuals or the wrong subset of individuals, and for the genetic situation, this will lead to then changes in the brain that we might describe as autism in terms of the clinical manifestations of those changes. They also mentioned that some GI tract dysfunction may arise from this particular pathway because of the intimate relationship that the immune system has with the gut in terms of oral tolerance and the idea of the immune system be able to determine whether or not what we're ingesting is going to be harmful to us. So this is the first model that they propose, and this is this direct cause and effect relationship between the immune system and the developing brain. In the second model, they propose that a toxicant could impact both the developing immune system and the developing brain. And so this is not specifically a an effect on the immune system that leads to an effect on the brain, but would be a concurrent effect. And again, this is going to be in part dependent on the genetic background of the individual who is getting exposed. So this is a little more of a complex model, because then it tells us that two different systems are being altered by the same toxicant. And there may be crosstalk between the systems, but the alteration inflicted is by the toxicant on each system independently. In a third model that they proposed, there's also going to be some independent but associated adverse responses to a particular toxicant. And this is even more complex, because Dr. Dewitt and his partner brought in this idea that the agent could produce these parallel multi-system dysfunctions, so that the comorbidities that we often see in children diagnosed with autism are from environmental toxicant effects on systems that are being impacted independently. So this model reduces the degree of crosstalk among the different systems and suggests that when we see changes in different systems, these are independent events, but the commonality is perhaps the timing of the event, of the exposure, or the underlying genetic background of the individual who's getting exposed. And so these are all presented in this 2008 paper by Dewitt and Dewitt. Now, we all know Dr. Ashwood and Dr. Van de Water through their interaction with ARI and the various webinars that they've presented. And I'm giving a call out to both of them, because I've been following both of their careers for many years, because in my opinion, they are really pioneers when it comes to uncovering some of these immune linkages in ASD and other neurodevelopmental diseases and disorders. And they've really done some amazing work to identify the role of some maternal cytokines and chemokines during gestation in terms of autoimmune damage that might lead to changes in the brain. They've also helped to identify specific immune signaling molecules and pathways, including information from the gut to the brain, and some work with microglia, the cells that I'm going to talk about today, and how they might be impacted. So I think that a lot of the work that I've done in terms of immune dysfunction and the contribution of immune dysfunction to changes in the brain come from the work that these two amazing scientists have done. And no, I don't have anything that I expect either of them to review anytime soon. I'm saying this because these scientists have been inspirations to the type of science that I do in my laboratory. But whenever I see any sort of publication or information on the relationship or parallels between the immune system and the brain, my question is sort of that chicken and egg question. What comes first? Is it the immune damage? Is it the changes to the brain? Are these independent events occurring at the same time during development? And so when I set up my research questions in the laboratory, these are the questions that I'm hoping to understand and explore in a little more depth. So which came first, the immune dysfunction or the alterations in the brain? So I'm going to define for you developmental immunotoxicity. And developmental immunotoxicity occurs when exposure to an exogenous agent skews the immune system away from a normal immune response. And it can skew in one of several directions. So when we talk about immune dysfunction that arises from developmental exposure to a toxicant, we might be talking about immune suppression, such as suppression or a reduction in the amount of antibodies necessary to mount an effective response when a vaccine is given, for example. And some of the contaminants that I work with that are known as per and polyfluoroalkyl substances have been associated with a reduction in responses to vaccines in children who have elevated serum concentrations of these compounds. We might also talk about allergic disease being a form of developmental immunotoxicity, and that would include asthma. So having sort of a hyper-responsive immune response as opposed to an under-responsive immune response. And I think we've generated a lot of data over the years that demonstrates exposure to environmental agents can make the immune system go a little haywire, so that we have responses to antigens that we wouldn't normally have responses to. I know living here in Eastern North Carolina, my allergic responses have increased dramatically over the past years. Another type of misregulated immune response would be autoimmunity, and this is when the immune system mistakes self for non-self, and so our immune system starts to develop reactions against our own systems and our own tissues and our own cells. And then finally, misregulated inflammation or inflammatory disease. This is really emerging as one of the greatest manifestations underlying a lot of chronic diseases that we see emerging in pediatric populations. Dr. Dewitt and some other colleagues have published a number of papers looking at chronic diseases and how they emerge during childhood and then grow as the individual moves into adulthood. Many of these diseases have an inflammatory component, and while inflammation can be healthy as an acute response, when inflammation is chronic, we know that it can lead to some persistent changes in how our body responds to other events, and it may even exacerbate those other events to the point of a disease or a disorder. So I'm going to talk about these just a little bit individually in terms of what we might say with respect to autism. In 2012, Rosenthal and Fry, and I think those are names that are probably familiar to all of you, evaluated a set of data and did sort of a systematic review of the data related to autism and determined that immune dysregulation and inflammation along with oxidative stress were areas of significant research growth in autism in terms of the number of papers published. Now, number of papers published then is indicative of the type of research going on. And so from this, we can conclude that this area of research has grown because the number of relationships have grown and the depth of our understanding related to immune dysregulation in autism has grown. Dr. Dewitt in 2012 also, again, concluded that misregulated inflammation is perhaps an underlying condition that underlies most, and he was even as bold as to say, if not all, chronic diseases and conditions. And so we're starting to understand more how immune dysfunction can be connected to many types of diseases that we might experience throughout our lives, not just during childhood, but that may emerge during childhood. A paper came out just recently by folks from Dr. Ashwood's and Dr. Vanderwater's laboratories, and this is again a paper that's looking at maternal anti-brain autoantibodies that are able to make contact with the fetus during gestation and may be a risk factor for the development of ASD and other types of neurological disorders. And I won't go into this into a great deal of depth, as Dr. Vanderwater gave a webinar to update the community on the type of research she was doing in this area just last spring. In terms of allergic diseases, a study just recently came out of Italy where, again, the authors did sort of a systematic review of the literature looking at maternal allergic, oh, sorry, wrong paper. That's the 2017 paper that Tonacci paper is the systematic review where they actually looked at autism and asthma, and they determined that there were some associations. The paper from 2018 that just came out was from Dr. Ashwood's lab and also, I believe, work by Dr. Vanderwater, where they were looking at a model of maternal allergic asthma. So this is a mouse model of maternal allergic asthma, and they found that there were some epigenetic changes or changes to how the DNA expresses or makes proteins, but not changes to the base pairs of DNA directly. They found that these epigenetic changes were detectable in microglia from the offspring of these mothers that had allergic asthma induced in them, and that these pointed to changes in immune signaling, changes in microglial inflammatory signaling and regulation of microglia development. So their conclusion was that maternal immune activation through their model of maternal allergic asthma could alter epigenetic regulation of microglia, which then in turn could lead to changes in the developing brain. And this is a very new paper in a journal called Glia. So I'm going to focus a little bit on this very first model. So in my laboratory, we're looking specifically at insults to the developing immune system by environmental toxicants. And very often we look at these emerging environmental toxicants, or toxicants that we know or expect to be in drinking water, but for which we have no current regulation. So these are a concern all over the United States because we don't have regulations to ensure that we're not getting exposed to them. So in this particular model, we're using an agent that we know is going to perturb the developing immune system, and then we look at changes in the neuroimmune system and how these changes in the neuroimmune system might lead to changes in the brain. And when I say neuroimmune system, I'm specifically referring to a group of cells known as microglia. And these are the brain's immune cells. They're tissue-specific macrophage, so they are cells of innate immunity. And for a long time, we thought microglia were just the housekeepers of the brain. They clean up stuff. When there's some damaged neurons, they come in and sop up the mess. But we're discovering now that microglia are dynamic and are involved in many different homeostatic, protective, and developmental functions in the brain. But let me tell you a little bit about our model first. We're using a double hit or a push model. So we're using a particular rodent model that has a genetic vulnerability, so it has specific mutations in it so that there's a higher likelihood it will experience pathologies of a specific disease state. And then we're using an environmental toxicant in combination to ask questions about whether or not particular severity or onset of certain pathologies increases. And this is where I'm going to talk about Alzheimer's disease. So we're asking questions about Alzheimer's disease to understand early life immune dysfunction on brain alteration. So yes, we're asking questions about a disease that we typically associate with aging, but very, very early in development. So we're asking, if we expose an Alzheimer's disease model to a toxicant very early in development, does this change the trajectory of the disease? So we're doing this so that we can understand a little bit about how microglia change what they do in the brain over the course of an organism's lifetime. And I want to point out that the remaining slides, for the most part, come from work that Dr. von der Ems did here in my laboratory for her dissertation. The work has been some of the work has been published in a paper, and that is in the Journal of Developmental Origins of Health and Disease, just back in 2017. And then she also presented data at the 2017 and 2016 Society of Toxicology annual meetings. So I won't be presenting any data that she hasn't presented or published in some way, and I'm hopeful, if she's here watching, that her second paper is well on its way to being submitted. So we'll see if I get an email from her about that. So Alzheimer's disease, and again, this is a disease that most people are probably fairly familiar with because it's been discussed in news and in scientific papers for a long time, but maybe not so much. My uncle passed away from Alzheimer's disease a couple of years ago, and when he was diagnosed, my aunt said, well, this is the first time I've ever heard about this disease that he has, this Alzheimer's disease. And I consider my aunt to be fairly aware and educated, but she wasn't aware of this disease that could strike people as they age. And so this is a progressive, late onset type of neurodegeneration. Individuals who are diagnosed have sometimes slow, sometimes more rapid cognitive decline, memory loss. There can be some changes in personality, and then all of these may work together to result in a loss of autonomy, so that many people in the end stages of Alzheimer's disease will need around-the-clock care. There does seem to be some data that suggests that there's a higher prevalence and a faster decline in females, but we haven't really figured out why. And Alzheimer's disease is characterized by some hallmark molecular pathologies. So there's a buildup of an intracellular protein called tau. So the neurons get a buildup of tau, and they become these little tangles that we think inhibit or interfere with the ability of neurons to do what they need to do in the brain. There's also a buildup of a protein outside of neurons called amyloid beta, and these are referred to as senile plaques. We think these also might lead to or exacerbate some other problems within the brain itself. And then associated with these tau tangles and with senile plaques, would be region-specific neuronal death. And there are certain regions of the brain that are more rapidly and affected earlier than other regions of the brain. So I mentioned microglia. These are the immune cells that live in the brain. And as I said, we used to think, oh, they're just macrophages that cruise around and clean up messes. But this paper by Bilbo and Schwartz, and Dr. Bilbo is a faculty member now at Harvard. She used to be at Duke University. This is a really nice figure because it tells you about the very amazing roles that microglia play in the brain. So they don't just clean up messes. They help to support synapse formation. They help to prune dendrites and synapses during certain periods of development when pruning is going on. They submit factors that help cells to grow. They still do cleanup jobs, but they can be both neuroprotective and they can be, in some regards, damaging because they can secrete inflammatory factors. And in fact, a lot of the work that's being done with microglia really does tend to overemphasize the inflammation and the inflammatory component, and the work tends to overemphasize the damage that microglia do rather than the protective mechanisms that they employ throughout development and life. In Alzheimer's disease, microglia may play multiple roles. So they may be subject to some of the genetic risk factors, so they may be predisposed to becoming dysfunctional in how they work and maybe increasing the amount of inflammatory signaling that they do. And then they may also have impacts on the brain itself in terms of how they respond to various agents that are being produced during the progression of Alzheimer's disease. And so some of what Anna tried to do with her dissertation work is to understand what's going on with microglia in Alzheimer's disease, especially when there's an early life immunotoxicant. Also some work done by Dr. Bilbo and her group demonstrated that there's sexual dimorphism in how microglia develop, and that there are differences between males and females during development in terms of the morphology or phenotype or shape that microglia occupy during development. So microglia can be ramified or amoeboid, which means that they're sort of, when they're ramified, they're sensing and sort of taking in all sorts of signals to figure out what they need to do next. And when they're amoeboid, they're in that more classical macrophage, let's get some work done morphology. And so we can try to understand what's going on with microglia by looking at what they look like and also understanding some of the signals that they produce during development. So our global hypothesis for this particular project is that early life immune dysfunction induced by exposure to an exogenous agent in combination with a genetic predisposition will increase susceptibility to later life neuropathologies. Now, in this particular experiment, we used a mouse model that is a transgenic mouse model, and it's known as a 3xTG-AD model. This mouse has 3 specific mutations so that it develops these tau tangles, amyloid beta plaques, and cognitive declines fairly early in life, between 3 and 9 months of age. When we exposed these mice to lead acetate in their drinking water, we gave it to them orally, and we used a very low dose. And it's a dose that we knew that they could tolerate, but also a dose that's been associated with some immunological effects. And we did the dosing between day 5 and 15 of their postnatal life. This is a period of development that's important for microglia, because, as well as for neurons, as microglia are migrating and moving into the brain and getting those tissue-specific signals that they need for differentiating into these brain-specific macrophages. Now, we were looking specifically at whether microglia were active, so in a state where they're actively looking for things to clean up and looking and secreting agents. But in this particular experiment, we really only distinguished ramified from amoeboid microglia. We didn't ask questions about what type of activation state they were in this particular experiment. And in this particular experiment, Anna demonstrated that we saw an increase in microglial staining density in male animals at postnatal day 50. Basically, what this means is that at postnatal day 50, males had a greater number of amoeboid microglia relative to females. So we interpreted that as males having more activated microglia than females at postnatal day 50. And when we looked at females and males and compared them across time in this transgenic mouse model that was exposed to lead, we saw that females had decreased microglial staining density relative to males at all time points. There was some variability associated with lead exposure, but this told us that males were having this heightened inflammatory response at postnatal day 50, and females were not. Females had a lower activated number of microglia throughout their lives compared to males. We also evaluated these data by flow cytometry, so a different technique, and what we saw was the same thing. We saw that males had this increased number of microglia at postnatal day 50, whereas females did not. And so this is just a different way of looking at these data. We also looked at amyloid load, and what we saw in females and differences that we saw with females and males was pretty interesting. So we saw that at postnatal day 50, males had a much lower number of amyloid beta plaques than did females. And so remember, this is then going to be correlated with that increase in activated microglia. So something was going on with the microglia at postnatal day 50 in these males that was associated with this decrease in amyloid beta. Now, if we look at postnatal day 90 and 180, we see that the amyloid load is about the same between males and females, but there was a difference at postnatal day 50 that we think was associated with how the microglia were behaving. And so we saw from this first set of experiments that microglia can be activated, and there can be a difference between males and females in terms of activation. And one of the conclusions that we made is that males were protected by this activation at a younger age because they had reduced amyloid beta relative to females. And we also can see that females lack this activation at this younger age, and then they had greater, not necessarily statistically, but greater levels of amyloid beta throughout life. So this early life exposure altered the way that microglia responded to amyloid beta and other changes in the brain later in life. Now, I mentioned that microglia can be bad if they're activated in a certain way. And let's go back to this figure. We know that microglia can exhibit two types of activation state. And this is a definition that we've come up with for macrophages. So microglia can be M1 activated, which means they're going to be producing more cytokines that we would consider pro-inflammatory, or they can be M2 activated, and where they would secrete cytokines that we consider to be more neurotrophic or neuroprotective. So M1 is generally regarded as bad microglia because it's pro-inflammatory, and M2 is generally regarded as neuroprotective. But that's not necessarily always true. That is dogma that's been associated with microglia from work that people have done with other types of macrophages. And remember, microglia play many different roles during development, so there may be all sorts of microglia are interacting, whether they're activated M1 or M2 or not. And remember, not all inflammation is bad, because inflammation can be a beneficial response to an acute exposure. So it is a necessary response, but when inflammation, as I mentioned, is chronic and maintained over time, that's when we tend to see the more pathological changes that we associate with an inflammatory condition. And in experiment number 2, we added in a wild type mouse. So we still had these transgenic mice, but then Anna added in these wild type mice so that we had a control. And she reduced the dosing period from 5 to 15 days to 5 to 10 days postnatal, so that we would have a slightly narrow window and a better idea of the role that microglia were playing during a time when certain microRNAs were being expressed. I'm not going to share any of these data with microRNAs because Anna hasn't published them yet, but she was interested in certain of these microRNAs, which are regulatory RNAs, and how they were communicating with microglia and controlling microglia, and then the communication that microglia had with neurons. In this particular experiment, we looked at different surface markers for microglia so that we could distinguish through flow cytometry whether microglia were more skewed towards an M1 or pro-inflammatory phenotype, or M2 or more neuroprotective phenotype. And let me tell you, Dr. von der Ems is brilliant, and she put all of these protocols together, and she did all this wonderful research. And I think one of the things that I'm going to miss most about her is her coming into my office with papers and saying, we need to do this, and here's my justification why. She builds these wonderful thought puzzles and always had every single one of her steps justified. So I'm really going to miss that about having her in my lab. So in the second experiment, as I said, Anna was trying to determine the difference between M1 and M2 skewing for microglia in these different models. And the main point to take from these different figures is that when wild type mice, these mice without mutations towards Alzheimer's disease, were exposed to lead, they had higher M1 markers at the earliest time point. So they had this, what we would say, appropriate inflammatory response earlier in life, that postnatal day 120, where there may still have been some changes going on from the lead, but as they aged, these responses sort of normalized to the levels of unexposed mice over time. But if we look at the mice with the mutations, and when they're exposed to lead, we see that they had increased markers for these M1 microglia that persisted over time. But what I want you to notice in this top right graph is that female mice at the latest time point also had increased M2 markers at postnatal day 240. So the female mice didn't have this response that we saw in the male mice earlier in life, and then they have this differential response later compared to males later in life. So this is giving us some clues about how maybe males and females differ in terms of their response to a toxicant, and then how their microglia function as a disease progresses due to genetic factors. Something else that Anna looked at is whether or not microglia are doing what they're supposed to do in terms of neuroprotection. And one of the things that they do is to sequester potentially toxic agents. One such toxic agent that can arise from certain types of metabolism is iron. Yes, iron can be toxic. And ferritin is a protein that is an iron storage protein in microglia. And so these microglia help to store extra iron through ferritin. And you can measure increases in ferritin as a marker of iron sequestration. And these sections are from a region of the brain known as the hippocampus, which is a region of the brain that is preferentially targeted in Alzheimer's disease. And what Anna demonstrated is that that in combination with this M2 microglial phenotype skewing that we observed in these older female mice, we saw that they didn't sequester as much iron as the male mice. We saw that they weren't really doing the job that they were supposed to be doing. And our conclusion is that this is a marker of microglial dysfunction. So not only did the female mice have an inappropriate response early in life, when they had an increase in M2 marked microglia later in life, these microglia weren't doing what they were supposed to be doing. They weren't appropriately storing iron, whereas the male mice's microglia were appropriately storing iron. And so these data really suggested that regulation of microglia activation, it can be altered by early life exposure to, in this case, lead, and that these changes persisted into adulthood, and that neuroprotection was impaired as signs of Alzheimer's disease developed. And I'm going to share with you one of my favorite sentences that Dr. von der Ems ever wrote, and I did paraphrase it a little bit, but she said, "Therefore, typical pathologies associated with Alzheimer's disease may represent failed compensatory responses and maladaptation rather than aging-related senescence." So one idea is that, well, as people age, their microglia age and they don't do what they're supposed to do. But her dissertation data demonstrated that this early life event changed the capacity of microglia to respond appropriately later in life. And so just let me remind you of these critical windows in development. So these are going to be critical windows of development for maturation of microglia, as well as critical windows of development for neurons and synapse formation. So if we have microglia already starting to be altered early in life, that tells us that they might also alter the communication between microglia and neurons also might be altered, leading to very early changes in the brain that in some people might exhibit or present as autism, and in other people as they age as Alzheimer's disease. Now, I'm not saying that the exact same pathway for autism and Alzheimer's disease is the same. I'm just saying that microglia play a very important role in the brain during development and throughout life. And so there can be similarities in terms of the experience of microglia during development that lead to these changes in the brain. Now, there can be additional critical windows of development for microglia and for neurons throughout life. You know, puberty is a very sensitive period of development. There are changes that go on in the brain. Microglia can respond to hormonal signals during development, and we can see changes in the brain at adolescence due to these changes in microglia. So even though we tend to focus on the gestational and early postnatal period, there also are factors later in development that can be impacted by external influences because they are these critical windows of development. So let's go back to that 2008 paper. Dr. Dewitt and his partner identified 4 primary landmarks of immune development that represent critical windows for both developmental immunotoxicity and autism. And it has to do with immune cell seeding of tissues. So as those microglia move into the brain and get used to the neuronal microenvironment, that would be an example of one critical window. Thymic events, so we have maturation of certain types of cells in the thymus, and events that take place in the thymus can be very important in terms of how these cells mature and develop. There's production, seeding, and activation of regulatory T cells, and these are cells that play, we think, a very important role in the regulation of immune cells that can be and they're associated with autoimmune disease. And then there's microglia and astrocyte, which is another type of cell that lives in the brain, activation and or dysfunction. And they said that not any one stage of development has been identified as a key window, but what these overlapping critical windows suggest is that critical windows of vulnerability may be at this neuroimmune axis. So looking at the immune system and how it influences brain development may be a very important avenue of research that we need to develop to get a better handle on neurodevelopmental diseases and disorders associated with development that may also lead into changes we observe in aging. And there are some correlates of Alzheimer's disease with ASD, and this may be related to failed compensatory responses and maladaptation that are associated with environmental agent exposures and interactions with genetic factors. So the possibility doesn't really narrow the research landscape. It actually makes the research landscape much more complex and much more confusing. But if we can target some of our resources at cells that have this common association with various types of diseases and disorders, then we might be able to get a better handle on what's going on during development when the immune system is affected and those effects lead to changes in the brain. Now, in terms of environmental factors that are common to developmental immunotoxicity and autism spectrum disorder, there are many that have been associated and explored in laboratory studies. Heavy metals would be one example, and these are from known effects on the developing immune system, and then associations that we observe in epidemiological study. We see commonalities for drugs of abuse as well as drugs of therapeutic value. Chlorinated compounds have been associated, as well as emerging pollutants. There's been quite a few papers recently on the role of particulate matter and nanomaterials and its association with autism. Endocrine disrupting compounds such as bisphenol A have been associated, as well as phthalates and polybrominated diphenyl ethers or flame retardants. It seems as if the list is getting longer and longer each year. But I think that this is positive, because then we start to get a better handle on what we might want to do to control or reduce our exposures. And then we can also start to develop interventions that might reduce some of our contaminant load as we start to think about having children. So in terms of future directions here in my laboratory, I'm actually working on another type of model associated with Gulf War illness, yet another type of neurological pathology. And what what I'm working on with my colleague, Dr. Lakshmakant Deshpande at Virginia Commonwealth University, is trying to correct these dysfunctional microglia and trying to correct neuroimmune crosstalk. And our hypothesis is that if we can correct this crosstalk, we might actually be able to reduce persistent symptoms, even though in Gulf War illness, the pathologies are already there. And so we're exploring this particular model and are anxious about hearing about funding starting, well, in February or March. We should find out about a particular grant proposal that we've submitted. So I'm still moving forward with trying to understand the role of microglia in various neurological diseases and disorders. And at that point, I'm going to conclude. I wanted to make sure that I left about 15 or 20 minutes for questions, and so I welcome your questions, and thank you for listening.
Great. We've got lots of questions. So anybody who's listening, if you've got a question, you can type it in the question section now. I'll start going through the questions that we've already received, but feel free to add more questions if you haven't had a chance to type one in yet. The first one, this one is about environmental toxins in the environment. And I know you touched on it just briefly in your last slide, but they're asking about what are the, I don't know if you could even begin to make a brief list, but if you were to select 4 or 5 things that you would avoid in the environment that are very common, what are some steps people can take to protect themselves from any toxicants in the water or other common exposures?
Well, I think some of our strongest data have come from heavy metals. So we do have some strong data suggesting that heavy metals have been associated with both DIT, developmental immunotoxicity, and autism. Unfortunately, because these are considered sort of legacy contaminants, we have measures in place to ensure that these are not going to be in our drinking water. Of course, these measures sometimes fail, as we learned from Flint, Michigan, but certainly checking your local water utility. If you have public water, they will report unregulated contaminants as well as some unregulated contaminants. You can also have your city or county health department test your water if you're on a well, or you can pay to have that tested. And certainly efforts to reduce your exposure to heavy metals is going to be optimum. And there are some filtration devices that you can use as well. But most heavy metals should be fairly easy to avoid through current technologies that we have. Otherwise, chlorinated pesticides have been associated as well. So if you can't afford to eat organic, eat organic. If you can't afford to eat organic, then try to eat fruits and vegetables that have been grown in the United States, because we know about the laws and rules that we have that regulate pesticides. So sometimes I'll see, oh, I need a yellow pepper, but it comes from a country where I'm not sure about environmental protection, and so I will substitute a green pepper that I can buy here in the United States. And there's also some emerging data for bisphenol A and phthalates and other types of endocrine disruptors and polybrominated flame retardants. And there are lots of alternatives that you can purchase that don't have these toxicants in them. The question now that we have to start asking, though, is, are the alternatives that are being used to replace any better? So I think there are some really nice websites, there are some green living websites, and there are some toxic-free websites that can help to direct you to foods and products that have a reduced toxicant load, so you can reduce your individual exposure.
Okay, that's great. The next question is about international research. So this person's asking about data regarding race and nations relative to microglial disorders. So are you aware of international research on this? Or I know primarily you work in the US, so I don't want to put you in on the spot, but we have a lot of people who tune in who are not in the US. So they're asking about that.
So one of the papers that I mentioned was from a group out of Italy. They were looking at the associations between asthma and autism. In terms of microglia, when I go to meetings, they're international meetings, and there are people from all over the world studying microglia and neuroinflammation. I can't rattle off the top of my head who the big players are in other countries. Well, I can mention Emanuela Corsini, who's at the University of Milan. She does a lot of work with emerging contaminants in the immune system in general. But if you have the opportunity to read scientific papers, look at where the journal authors are from, and you'll get a sense of who's doing a lot of work. There's actually a lot of good data coming out of China. We have a lot of the papers that I review that concern microglia are from some of the state key labs in China, and they're doing some really amazing work with microglia, and especially mechanistic work and signaling work with microglia. So they're very strong researchers coming out of China.
Okay. This next question's, are you aware of any clinical trials coming up to evaluate the effectiveness of immunosuppressive drugs on skewed innate immunity?
I am not aware of clinical trials. I should point out that the work that I do in my laboratory does not involve clinical work. I don't work with clinicians, not for lack of trying. I don't work with human samples. I don't work with human subjects. So I have to admit that sometimes my knowledge of clinical trials is not as strong as it could be. And right now I've really been focusing a lot of my work on these per and polyfluoral alkyl substances. And they've been taking up a lot of my time right now just because of their emergence here in North Carolina. But certainly I can look that up and get back to Denise so that she can share that to whomever asked the question.
Okay. This person's asking about, I think you addressed this in your talk, but I think it's an interesting question. They're talking about severity being more intense in early childhood to exposure, or would it not matter at what point? So you talked a little bit about how the brain continues to develop throughout the lifespan. So would it be logical that what happens in utero and immediately after birth would have a much bigger impact? Or would you suspect that throughout the lifetime there could be?
Yeah, I think it really depends on what particular events are going on at that particular point in time. So if you remember back to one of my figures, let's see if I can pull it up. Well, here we go. So the particular figure that we have eventually right here. So we can see here that this is the difference in terms of microglia between males and females, and the peaks would represent activation numbers. So there's higher activation for males earlier in life compared to females. And when we look at what's going on in males at that particular point in time, at least in rodents, there's this very early surge of postnatal testosterone. And we think that this is involved in helping to lead to sexual dimorphism in the brain that is specific to males. And it's associated with an increase in microglial numbers. But there are also other critical windows associated with particular events. There are certain windows associated with a high degree of synaptic pruning. And if there are impacts on microglia during that point and they're dysfunctional, then they may not prune back an effective or normal number of synapses. And there's been some research coming out to demonstrate that children who have been diagnosed with autism have a greater number of neuronal connections. I think there's some functional MRI data suggesting that there's an increase in these connections, which leads to this increase in sensitivity to environmental stimuli. And so when I see data like that, my first thought is microglia weren't doing what they were supposed to be doing at that particular time. So sensitivity, I define sensitivity as what's going on at the particular time. So what's happening in the body, when is exposure occurring, and is there a correlation between what's happening and when the exposure is occurring? So there's greater or lesser degrees of sensitivity for various systems and events in the body.
Okay.
I hope that sort of answered the question.
Yeah, I think it does. The next question is about inflammation, which you talked about quite a bit. And you talked about how they can both be protective and they can also be damaging. So this is a question about testing and detecting these impacts in humans. So I know that you're a researcher, so I don't want to put you on the spot in terms of clinical analysis, but are you aware of testing that people do that analyzes these different inflammatory factors in the brain?
I don't know in the brain. I mean, there are some serum measures that we can make, and in humans through clinical evaluation that tell us about certain markers of inflammation. These tend to be more associated with cardiac health, but there are markers of inflammation. The question is, though, when you see, for example, an elevated marker of inflammation, is that inflammation associated with an acute or short-term event, or is that a chronic, long-term, low-level inflammation? Now, we can measure these signals very easily in cells and in animals. It's a little more challenging to evaluate in humans because, again, there's that question of timing. So inflammation taken at one point in time without understanding what's been going on is going to be very difficult to interpret. So you may be able to get a clinical picture of inflammation at a particular time, but I don't think it's going to be extremely helpful in letting you understand what's happened over the course of development or over lifetime. Unless you're a sample in my laboratory and I can manipulate you, but
right. I know that's a tough one to answer because you're right.
Yeah.
It's almost like you'd have to have post after death tissue, right? To get it.
But there, and I think there are some improvements in some of those clinical measures being made as we speak, and I think some of our doctors are getting a better understanding of how to interpret some of those clinical measures. But still, the timing issue is the big question.
Okay, the next question is about using rodents for trials. So obviously humans and rodents are very different. When you're making hypotheses and measuring those things, how do you can take that into consideration? What are some things that researchers think about when they're using rodents as a model for human research?
Well, so in terms of what we do in my laboratory, I don't so much think about the rodent as being a model for humans. I think about the rodent as being a model for a particular pathology that we may observe in humans. So for example, this 3XTG-AD mouse has mutations that are the same mutations in humans that are associated with early onset familial forms of Alzheimer's disease. Are they going to develop the exact same disease as humans? Absolutely not. But these mutations lead to similar pathologies in the brain. So these mutations lead to increases in amyloid beta. These mutations lead to increases in tau protein. These mutations lead to neuronal death in specific regions that we can associate with changes in behavior. And so when we look at microglia in these particular models, we can ask questions about how microglia are responding to these pathologies in the mice that also occur in humans, but there's not this one-to-one comparison. So we're often very cautious about overextending what we observe in mice to humans. So we say we see these observations in mice. This is an amyloid beta mediated effect. We also know that amyloid beta plays a role in humans, and so then it would be, well, do microglia also play a role in humans? But we really can't answer that question without human samples and human tissues. So a lot of what I do in my laboratory is trying to understand the basic physiology in a rodent model, and then somebody who I might work with who works with humans would make that leap.
And you touched a little bit on vaccines, and I've gotten quite a few questions about adjuvants. So
I think this is one of those questions that I think is very challenging to answer because of some misinformation that has gotten out, and I think some of that misinformation has become sort of the way we think about vaccines and childhood diseases. I don't work with vaccines. I do employ a particular model in my laboratory that is analogous to a vaccine. So I do look at what would be considered a vaccine response, but I don't specifically look at the role of vaccines or any adjuvants associated with vaccines in altering brain development. I do know that, I did write an editorial with Dr. Dewitt about a particular paper that did highlight vaccines as a potential association with autism, and our response was that the data are very, very sparse, and that from an immunological perspective, we don't have strong data to suggest that vaccines themselves or the adjuvants used in them are strongly associated with autism. But again, this is not my area of research, and I haven't looked into this question in a number of years, so I'm probably not the best person to answer that particular question.
Okay, great. Thank you. One more question that we have, and I'll try to frame this. We can do this as the last one since we're almost out of time. One person's asking about any kind of correlations between socioeconomic status and exposure to toxins. I know that there is a wide difference in the autism community about who is diagnosed and who is not. The higher socioeconomic status a family has, typically they're more likely to get the assessments and get a diagnosis. But I am wondering if there is data that shows that there is a correlation between higher or lower socioeconomic status and exposure to toxins. Are you aware?
Absolutely. Yeah, there are huge differences, and it really depends on the toxicant. So some toxicants have a higher association with individuals who have a higher socioeconomic status because of the products that they're able to use in their homes. For example, certain products may have certain contaminants in them, and if these products are more expensive, then they're going to be purchased by those who can afford to purchase them. Other contaminants that are more prevalent in the environment, maybe in well water and in public water systems, are going to likely have a greater impact on those at the lower socioeconomic level because they can't afford in-home filtration devices or afford to get their water tested. Similarly, when we look at pesticide load, those who can't afford to buy organic foods or really even afford to buy extremely healthful vegetables may have a higher pesticide burden than somebody who can afford organic foods and to get food at a farmer's market, which actually is really affordable, but unfortunately for many, farmers markets are not as accessible because of transportation issues and where they're located. So there's lots of data on discrepancies in terms of socioeconomics, and one of those issues is called environmental justice. Some people refer to it as environmental racism, and that means that individuals who live in economically depressed areas tend to carry the greatest environmental toxicant burden, and those who are impacted the most tend to be children. A study was published in October of 2017 in a journal called The Lancet, and in this particular study, the authors noted that exposure to environmental pollution is the world's leading cause of premature death. 9 million people die a year from diseases associated with environmental pollutants. That's how many people live in New York City. And the greatest burden is borne by those who live in lesser developed countries and who are of lower socioeconomic status. And the subgroup that bears the greatest burden is children who are poor.